I have a question before everybody else. You described the different BSO classes. Say I go to Algeria and I collect a mouse. So we have the blanket permit.
Yeah.
How can I, as researcher, be certain that that bloody mouse does not contain the next COVID-5?
You can't.
How should I work with that?
Generally, the advice for animal or human tissue, if it looks healthy and you have no reason to suspect that it's not, and it's not known to carry anything which is dangerous, then level 2 will be the baseline. But you should do a risk assessment, of course, and make sure that that is the case. But if you have no reason to suspect otherwise, and then it will be level 2. There are some exceptions, of course, if you are importing somewhere from areas where they have, for instance, zoonotic or epizootic outbreaks, then, of course, that shouldn't be handled by you at all. And if you absolutely need it, then you can send it to the Swedish Veterinary Institute or something like that. Contact them, because then you should work at a higher level. Is that fair enough? Level 2 hours.
Thank you. Yeah, that's fair enough. So every time we import anything, an insect, a mollusk, anything else, we get in touch with you and you send the permit.
Yeah, if you need a permit, I will send it. There is a form on the website which you fill in and so on.
Thank you so much. Yes, Joanna, can you catch? Or would you pass it on?
Hello. I just want to understand, so everything that is not dangerous, like some cultures of algae from lichens, are they anyway level 1?
If they are not dangerous? Or
Yeah, or they are not parasites, they are just small algae living in symbiotic organisms. Are they level 1?
They sound like level 1 to me.
So everything is level 1 if it's not the other levels?
Yes, sort of. But then also remember, think about the animals. What happens, or
They're not
if it's, because it could be completely benign to us, but then highly pathogenic to other species. So just consider that aspect. But apart from that, I would say it's level 1.
So these are not animals, they're lichens, they're fungi algae.
Yeah.
Okay.
But of course, that, with the exception then, if you're bringing in something which could be a plant pathogen,
Yes.
that of course you shouldn't treat as level 1, right?
Yeah.
And then you should have a separate permit for that.
But if we get them like from another country now, sent by post, we
Yeah.
need to have a permit, even if it's level 1.
You would say, actually, if it's not pathogenic for humans and it's not an environmental issue and that could infect other species, then I don't think you need it.
Okay.
But there is also, talk to the transport company, because even if you don't get into trouble with the legislation working with biorisks, the transport companies have rules for if you want to send biologics, right?
Yes.
Yeah. So that could be another aspect where you
Thank you.
could come into trouble.
Speaking about the courier companies, so I have a species that I started the CITES legal work in July last year. The company has delivered it in Sweden, but the wrong way, so I still cannot use the samples up to now. I cannot extract DNA, and that's endangered species from Israel.
Okay.
Yeah, so courier companies, be careful. Do we have any other questions? Yes, if you get the mic over there. Actually, now we can open the floor, because that would be the last presentation before lunch, and we can open floor also for Sebastian, for you one. If you have Nagoya questions or anything legal, just go for it.
I don't have a specific question, you're more like your thoughts. Say you're taking soil and you're enriching or isolating species from that. Theoretically, you could get a plant pathogen or something else. Like, how do you properly handle when you're working with something you don't know?
Well, you would have to start with a risk assessment to see what do they have in this country or in this area where you're importing this sample from. And if it is something which is potentially a biohazard, then treat it accordingly. I mean,
I guess the question.
it is a difficult question, but you can approach it in a risk assessment in a way to handle it in a reasonable way, I think. And if you have any questions if it's reasonable or not, you can contact me and I'll see if I can help you out.
Okay.
Or
I can say something on that. So.
yep.
So first of all, you will have to have a permit, and you may not be able to get a permit to import soil as is into many countries. And second, if you do have a soil and working with a soil, the disposal of it has to be like a disposal of not the biological waste, but a special way of disposing the soil. It has to be burned basically afterwards.
But is this for all countries importing into Sweden?
That I cannot really answer. I don't think so. It depends. Where it comes from and what are every country's regulations. So one of the examples is all these plant parasitic nematodes. Every country would have its own rules, and most likely you cannot bring soil from Netherlands to Sweden without getting into big troubles in that. It has to go through the quarantine, well, screening, if you really want it to.
Okay.
Thank you. I go to Mafalda, and while I go to Mafalda, I can share other kind of experience. So before working with genomics, I was working with plant pathogenic fungi. So I had sometimes to import plants from other countries. So for that, I needed a special permit from Swedish Board of Agriculture, Jordbruksverket, where I would specify exactly what are my safety precautions in the lab. And that has been signed and presented at the customs. So that also takes time. So plan like 2 months before you actually go and collect your plants. So you contact the Swedish Board of Agriculture to get that permit. Mafalda?
Thank you. Actually, I have some questions for the Nagoya panel, and maybe it was for Nicholas. I'm not sure if it's still there, but my questions essentially, I just wanted to clarify, for example, because it was mentioned that it's not, we're not sure yet what date applies, for example, for something in the museum that was collected 100 years ago. Is the date of collection what applies, or the date where I sample for genetic or for sequencing that applies? So is that not clear, or is it that's because there's not a precedent yet, legal precedent yet? But does in that case, would precedent from another country apply? And I have another question after this one. I don't know.
Yeah. I don't think Niklas is with us, so I may answer that. What I would do is, like, first of all, see if the collector or like the museum or whatever institution there is that has the material, do they have some information when it comes to third-party use or anything about this specific material that you're going to use, and see if you can find anything there. Maybe it says like it's okay to use within research or whatever. Then I would say you don't need to care about the Nagoya Protocol if you can get some kind of okay green light from the institution. If it doesn't say anything, then perhaps try to see if does it say where the material comes from at all. And if it does, go to that country and see, do they have anything when it comes to material collected before 2014? Because some countries may still find they have a right to regulate it or not, and you might find some information there. If you don't find any information about it not in the museum, or you may not know where the material actually comes from, you only know it's collected before 2014, then I would say make an own assessment and motivate. I just say in the due diligence that this is collected before 2014, and therefore Nagoya Protocol is not applicable in this case. And then you have made your kind of due diligence.
You mean it's writing myself a document where I say I've done this research, and
Yeah,
to the best of my knowledge.
yeah.
Does this need to be assessed by someone at the university as well?
No, not really. I mean, you can write it in a document or somewhere where what you can access, but it's more like you need to be, if you need to show something someday later. For example, as I mentioned, for a funder or for a when you're going to publish it, or if Naturvårdsverket is making compliance checks, then you can kind of explain why, or you can say, here's my due diligence, and my conclusion is that the Nagoya Protocol is not applicable in this case because of this, and then you're agreed, I would say.
And then my other question was, at some point it was mentioned there's a mutual agreement document, MAT or MTA,
Yeah.
and the presentation says someone needs to sign that agreement, and so far I've been the one signing it, but someone at the university needs to sign it. Okay.
The legal authority of your institution. So in the museum, it will be science director, and the university it will probably be somebody head of science division, somebody who had legal rights to sign, or maybe legal department. Depends on, like in my museum, we do a kind of legal department. So in our case, it's science director of science.
Okay. So not me who's assessing the collection, but
No.
okay.
But I can also add a little bit to the previous question.
Take microphone, Sebastian.
We also kind of missing the issue of the date of export may also play a role, because if the sample was collected 100 years ago and exported from the country, that can also be interpreted as the date of access, and then you're fine sequencing it. But if a sample was collected 100 years ago and stays in the country, like Brazil, for example, it can be very, very old, but if you want to ship it right now, it's under Nagoya regulations.
Thank you. So the comment to Mafalda, the person with the legal signature or with legal signature right or signatory is usually the head of the department. So that is the closest legal signatory to the researcher. Right? I go further, and actually I have a question. So say the sample was imported by the researcher at SLU here in Uppsala, but they want to do analysis at SciLife Lab, which is Uppsala University. Do they need a third-party agreement they pick with a transfer to third party, yes or no?
Have a look on the agreement with the country where you have the permit. Maybe it says that it's not. That's a typical thing that they normally want to regulate. They don't want it to be free access to third parties because that is how they use control of it, right? It's like it's pretty. If you're a country with united resources and you agree with, like for this example, SLU, then you still know we are dealing with SLU. But if SLU lend it out to Uppsala and then Uppsala lend it out to Stockholm, it's like it will go pretty fast. Then they have no control where's our samples and what research is done with our. So that is a typical thing that they regulate. You're not allowed to share it with other, but most of the times you write it like you need if you get their consent, it's okay. So that's normally how you deal with it. You say like, okay, we got these resources, we want to share it with SciLife Lab or Uppsala University. Can we because we want to do, for example, non-commercial research, right?
Yeah.
Then you get an okay or not okay, and if you get the okay, save that documentation. And
All right. So the situation could be following. So for instance, this is an only case. So we have a sample from SOU. A part of the analysis is done at SciLife Lab Uppsala, and part is done at SciLife Lab Stockholm. Like when the researcher at SLU is deciding to do it in Uppsala or Stockholm or both, can it somehow reach out to the country and check if it is okay? And if it's not okay, what can be done at that stage? Say, yeah. So DNA in Uppsala and high C in Stockholm.
yeah, I mean, and typically when you plan your research, you kind of know who you will collaborate with. But sometimes, okay, you don't know, and it turns out later that we need the support of another university, right? Then yes, I would say. and especially as long as you're within the framework of what you're like got the okay to do, and you can explain, we need to get help here from this university. You should reach out, but it probably won't be any problems. I don't think so, especially as long as it's within the academia. I think it's more sensitive if you say like we want to share it with this company, that's a commercial company, because then you suddenly step out from the kind of academia world, where they may feel like, okay, but do we really know what they're going to do? It may turn into a commercial.
All right. So it's good for researchers to check before spreading the sample between different.
Yeah, if you haven't done that from the beginning because you didn't know that you were going to ask other universities to do some work
All right.
like yes.
Do you want to
UMTA, New Material Transfer Agreement. New Material Transfer Agreement.
between whom and whom?
All the parties.
Okay, thanks. If I have all the permits and everything is in place, and I have my genome, let's say I've only used it for phylogeny, but I have a whole genome data set, and then I want to put it out. Is it still, whatever happens afterwards, is it still my responsibility, or is it the one who takes the data, or was this the thing that is not out there yet?
I mean, you kind of touch a question here that is very, in my view, interesting because there is a kind of clash between the Nagoya Protocol and control of the genetic resources versus the open data, and both of these principles are, I mean, valid and worth to follow, but they not always comply with each other. I would say that if you collect the material, see if they're with the country they put any restriction on the material. Maybe they say you're not allowed to put it in any database after you sequences or whatever. Then you can't do it. But you could like at least propose to make it open data. I think that's also an important policy. But in the end of the day, it's the Nagoya Protocol is really based on that. All countries have decide over their own genetic resources within the country.
So it kind of follows also, do I then need, if I would use genetic material, like open genomes from a repository that have been collected from somewhere else, do I need to care if those genomes had the permits or no? No.
If you want to use it commercially and if your country requires a special permit for commercial use, then you get the genome from GeneVet, you still have to apply for commercial use permits from the country.
Thank you. Yes, but not for the basic research. Perhaps we can cut it out later, but from the recording. But I can say that in ERGA, we are putting together a group of scientists who would like to write an opinion paper about the hurdles that Nagoya Protocol is putting on the researchers and the amount of taxpayers' money and the time that we are spending instead of writing the research, and to present that, and hopefully legislators will perhaps come up with exception or something. But now, all that information that we put to you now, that Henrik presented, and Johan and Sebastian and Niklas, it's daunting, it's bureaucracy. We are scientists, okay, I'm not a scientist anymore, I'm not PI, but you guys are. So you would rather do your research instead of fighting like tons and tons of paperwork. So if we just sit there and be extremely compliant and said, yeah, okay, we have to follow, maybe it's time to sort of do something about that and make the authorities and bureaucrats know that's not perhaps the best way, because in their eyes, these things are new, and the discussions that we're having with DSI, and most likely they will not be under Nagoya, so that's a lot of work that is being put into that. But perhaps it's the scientists need also to educate the policymakers that some of the legislation is really difficult. and off-putting.
Can I just comment on that? I think you take up a very interesting thing. Niklas mentioned it in the end of his presentation, this DSI question that is very much debated right now. What I can say for sure that it will not be a bilateral system like we have with the physical samples. It will, if so, be some kind of, we are talking about the databases now, all the digital sequence information, and if it should be restricted or not. It will not be a system like the Nagoya protocol. Instead, it perhaps be some kind of subscription for the databases that you have to pay some money to access, but paying that money also means that you have no restricted access to all the information in the database. And like, this is just my personal view, but I can tell that I don't really think Nagoya protocol works as good as it was like meant to be when it was agreed 2010. There is not really that many agreements out there. There is not that much money flowing from user country to the country that have the Unity resources. So, and also with this DSI system in place that was designed as a parallel system, is like, isn't it? I mean, I can imagine that to avoid all the bureaucracy to make all these agreements that we talk about, why not just try to sequence it in the country where it is, and then I can download it from the database to make it more simple. I mean, that kind of practical way of doing will probably become more and more. I mean, that's how it works, right? You don't want to put a lot of work on bureaucracy if you can get it in a easier way. So
Thank you.
I think it will change a lot.
Thank you. And another question to the lawyers in the room. In Sweden, we have the minority, we have Sami, and they have the traditional knowledge. We also have allemanrätt. Perhaps you can explain, I don't know what is it in English, so you can go and collect things for non-commercial use. So what is the framework if a researcher going and collect something on the Sami land?
The framework is more nothing when it comes to Nagoya protocol, because Sweden has...
Not the Nagoya, but ABS and traditional knowledge.
Yeah, but that is part of the Nagoya protocol system, access and benefit sharing and traditional knowledge. And when it comes to that, there is no legislation. Sweden doesn't have any access legislation, so you don't need to care about those things when it comes to access and benefit sharing rules. So you're free to collect to do research.
All right. Thank you. Are there more questions? Yes, there are. Yeah, you have
It was mentioned before that you might need additional consideration if you're looking at genome function, this kind of thing. But say I'm just doing phylogenomics, and then I'm annotating my genomes, for example, and assigning the function. Like, where does the line sort of start where we actually look at gene function, for example?
Can you say that question again? I didn't think you're right.
Well, it's just, it was mentioned before that you might need more considerations if you're looking at gene function, for example, when you're sequencing. But if you're like annotating a genome, you're assigning putative functions, for example, and like where does it start where you might actually be concerned about additional?
Okay, yeah, I got it. That's a question about like what is using a genetic resources actually means, and that there is a whole like guidance document about that, where an EU document from the Commission guiding document what is seen as using a genetic resources, because we import genetic resources all day for consumption, right? Fruits and vegetables, whatever. That is all genetic resources, but it's not within Nagoya protocol. So when it comes to your question there, it's like, and that's a typical question I could get from my research. Hello, we're going to do this with these genetic resources. Are we in the frame, like, do we need to care about Nagoya protocol? And one of my first question would be, what are you going to do to be able to assess? Okay, are you using it in the way using is meant to be in the Nagoya protocol rules, or are you outside the scope? And if you're outside, you don't need to care about the Nagoya protocol, but you need to make some kind of assessment and as I mentioned before, it's also good to have it in written. But you could absolutely get help with that from legal unit or so on at NOS. But that's how you do it, obviously. And it's still you do the due diligence just by putting it clear that we are outside the scope of using it, so we don't need to care about Navarro protocol, and then you're done.
Thank you.
It's more of a comment from a marine biologist, but I don't know if you're aware, but more than half of our planet is international waters, so you don't have to apply Nagoya. You can. There's. Sorry.
Not yet.
Not yet. No. So I work essentially in the deep sea, but we have no paperwork whatsoever. So if you want to change organism and move to an organism that's in international waters, that's another maybe option, easier option as well.
Yeah, you're right, Pablo, but be aware of this BBNG regulation that came this year that will start to regulate also this kind of genetic resources. It will not be in the exact same way, because as you say, it's international waters, so you don't have any country to make agreement with, right? But there are other way of they're going to put this access and benefit sharing system into place also when it comes to international water. Yeah.
Hello. I've worked with unicellular eukaryotes, and usually when I go on a holiday to somewhere, I just collect some sand samples from the beach and just from the water to isolate them and get genomic data from them. But I was wondering that do I actually need something from Nagoya for such things, because I'm basically collecting beach sediments from the coast on a holiday and just from the water as well. And when I went to Poland, I found some samples for which we got quite a lot of genomic information, and it's useful for us. But then I looked for the Nagoya website, but there's no one really to email on Poland that, who should I email that, hey, can I have permission or something, because it was just collected from my holiday. And I never expected to find something. I just looked under the microscope and I was like, oh, this is interesting, and this is the sample, but what should we do in such cases?
Yeah, I mean, collecting things on your vacation at the beach, and no one will notice, and you can do it, and you can bring to your home probably. But it's more like if you find something when you do your screening, and this is really something that we could make some, do some research about. Then I would recommend you to, okay, where did I collect this thing, and do they have any legislation when it comes to Nagoya Protocol, or was it Poland?
Yeah, it's Poland. So one I collected in Poland, one in Italy, Australia, Thailand. I mean, pretty much my whole lab, wherever we go on holiday, we just collect something.
Yeah, and But I can tell you, all the countries that you mentioned, they definitely have some different rules on it.
Not Italy.
Huh?
Italy doesn't care.
No, Italy doesn't care, and I don't think Poland cares. But let's say Thailand, they do. So so you could. Let's say that when you do your screening and you want to do some actual research on the material that you collected in Thailand, maybe then it's time to start to look into it more in detail to see, okay, what do we actually need to get in order to be enabled to conduct the
But in the Nagoya website, they don't mention anything. We have already looked as much as we can, and there's no particular person that we could email and ask that, can we use this sample or not? Or
Do you mean this clearinghouse? Yeah. I can just mention it to everyone here. I mean that webpage is a good initiative, but don't expect all countries to reply there. I mean, it's a good idea, and I wish it worked better than it does. But yeah, that is what we have to deal with. When it comes to Thailand, I have the same experience. But when we did research there, we collaborated with the Botanical Garden when we collected the species. Maybe that's not the case when it comes to your beach. But let's say if it's within a national park or botanical garden, that would be my best to go if they don't reply from the ABS clearinghouse. Or if you have another good tips is to always collaborate with local partners in the countries where you, conducting research on the material, because they normally know more than you who to talk to or where to go.
Thank you.
Okay, hi. So I have barely dipped my toes in the field of genomics, but even I can tell that from 2014 to now, the field has changed quite a bit. So I was wondering if the Nagoya Protocol document includes anywhere like some clause or article that indicates how amendments or revisions can be made and whether the signatory parties can revisit the document every few years or like every decade or something like that if we as academics wanted to push for changes that reflect the rapidly advancing nature of the field and facilitate dealing with this bureaucracy. There's some way we can plan for that in the future, in the next few years or something.
Yeah. I mean, what it says, it has this conference of the parties system going on, the Nagoya Protocol. That is conferences that's going on year after year, where the parties meets and discuss what works and what doesn't work. And the question with the DSI that we mentioned about digital, how do we look about digital sequence information? Should it be regulated or not? That is a typical question that has been debated for a long time now. And yeah, so my answer to your question, absolutely, this is a living document that for sure gotta continue to develop. And as mentioned, I don't think Nagoya Protocol works in practice as it was meant to be when they signed it in 2010. And like what you should remember here, this is based on a principle that was found in this Convention of Biological Diversity. This ABS, Access and Benefit Sharing. And what Nagoya Protocol is trying to do is to put that principle into action. Because before Nagoya Protocol, it was more or less just nice words in this convention, right? But absolutely, it's a discussion whether the Nagoya Protocol actually does it happen in an efficient way. And my own opinion is no, but that may change, and I hope it will change, and with your input.
Yeah, I've luckily already gone through the Nagoya hoops together with Sebastian, actually. And what happened for us with our South African partner was that the first thing I realized was that Nagoya was written when people were really optimistic about the opportunities of genetic information. And then I think that a lot of countries, their officers kind of dropped interest because they realized that the economic value of genetic sequencing of mucus is not that great. So it has been a bit like drop in interest, which made it very hard for us to find a Nagoya officer in the country. And then what we realized was that, okay, so in this case, for our cow sequencing, then as long as we do not have any commercial intents, then South Africa, which I was working with, they don't require us to have any special provisions according to the Nagoya Agreement. The problem is, of course, if we find something and it becomes commercially interesting, then we would be in a difficult situation. So what we did in our case was that I'm doing the sequencing together with my South African colleagues. We do the sequencing in Sweden because we're really good at sequencing at side-by flat. So thank you for that. And what we do then is that we send the data back to our South African colleagues, and then we're doing the joint publications where we basically share all the DNA results openly. So then they get credit for those publications together with us. And then if someone manages to dig something interesting out of the data, then it's done in the normal, like Western standard way, where if you find something interesting and actually proves that it's relevant, then you can go through the entire innovation process. So that's how we shield ourselves from the ambitions of Nagoya. So open science actually really helps in this case.
Mm-hmm. Thank you, Thomas. So Nagoya originally was created for things like Ozempic. So that is the drug that was developed in Denmark, that actually it comes from a tropical lizard or salamander, amphibian or not, don't remember, but it was something with 4 legs and tail and like that. So the point is that the huge income that Denmark is making from Ozempic should be shared with the country where this lizard is coming from. But when it really comes not to the drug, but to DNA, exactly as Thomas says, that is, perhaps not so much monetary value as compared to the biopharmaceuticals or something like that. Do we have any more questions in the audience? Okay, Anna.
Just useful. Oh, thank you. This is more practical question. Let's say we are doing our homework. We look at. I have samples from different countries. I see what ABS I need next week, but then it takes a while to analyze the data, and things may change in half a year. What should I document that? This date, I found these results when I start to analyze the data, or is counting the publication, or like when you deposit the data. You know what I mean? Because you don't want to start a huge analysis and then you have to take out samples and do the whole analysis.
I would say it sounds like the most practical way to do it is to you make a note when you do the actual step, and if, as long as you don't hear anything else during the way, you can trust it is the same as when you collect it.
Okay.
That's how I would do. I mean, I don't want to say that all countries works this way, but most of the countries don't use like laws that gets started from an earlier date. If they come up with new rules, it's most of the time it's going to start a year ahead or 2 years later, and they will not start to apply it on material that was collected.
Yeah.
And like,
Thank you.
like.
Yeah.
I just want to comment something that we shouldn't bring samples when we go on vacation just for like biosecurity reasons, that we can be spreading diseases, like when you bring live organisms from places from other parts of the world, you may be introducing new organisms in your own country that may be causing new diseases that you didn't know that they could cause. Just these things.
Thank you so much. Thank you. That was an amazing session. I hope you all learned something new. And a huge thank you to Alexander from the Swedish Museum of Natural History for the insights about the soil and nematodes. Really appreciate it. And thank you, Sebastian and Ton, for being the main target and keeping the flag of the Swedish university lawyers.